Fabrizio is going to be the last speaker of the series of 2023, and we can say we're finishing with the icing on the cake. Fabrizio actually was graduated in the University of Trieste, then worked for 4 years nearby here. We are in Basovizza. He was in the other campus, and then he moved around the world, and then settled in one very prestigious institute on molecular oncology in Milan, when he's a principal investigator, and he's also member of the AIRC Institute of Molecular Oncology in Milan. Fabrizio published extensively in top peer-reviewed journals, and is the expert, or one of the experts, in age-related disorders. And this is very interesting, particularly for me, due to my age. And we are very grateful that he accepted our invitation. So the mic is yours, Fabrizio. You have 45 minutes or something like that, and then followed by some discussion. Thank you very much.
Again, thank you again. Apologies for the delay. Let me see if I can share the presentation, if this goes smoothly as we all hope. Do you see the presentation? Yeah. Is it full screen now?
It is full screen now. It's perfect.
Again, so as it has been mentioned, we are interested in the process of cellular aging, and more in general about organismal aging, but we are also a DNA damage lab. So I'll try to persuade you today that DNA damage plays a key apical role in the process of cell aging and therefore organismal aging. However, there is an opportunity to control the consequences of DNA damage, not DNA damage accumulation per se, but the consequences of DNA damage accumulation by targeting not the RNA, not the DNA, but actually the RNA. Hence the title of my presentation today. Probably we'll all agree that the disease can be broadly defined as a loss of homeostasis, a loss of equilibrium. And probably it's sufficient to think about all the conditions, and aging comes to mind, and the skin of this person when young or when old gives an immediate, very graphic representation of this loss of homeostasis. Importantly, however, aging is not just about wrinkles and lines on the skin, but it is, in fact, it's a serious risk factor for many important conditions, many important diseases, including cancer. That is the reason why we are studying aging and cancer in our. Aging is therefore important, but it's also very complex to study. And therefore, being cell biologists, we approach the problem in a perhaps reductionist way, and we are studying not aging in an organism, but aging at the cell level, because you could, again, simplify, reduce the complexity of this so complex question by proposing that we age, organisms age, because our cells age. And aged cells are also known as senescent cells, which are cells which are, by definition, unable to proliferate. They do not function properly, so they lose some functions, but they also acquire some additional functions. They acquire the ability to synthesize and secrete pro-inflammatory molecules, cytokines typically, which it's known as SASP for the senescence-associated secretory phenotype, and this causes frailty systemically, and of course frailty makes us more prone to diseases. Then the question becomes really, what causes this loss of homeostasis that, as I mentioned initially, is a cell loss of homeostasis, then becomes a systemic loss of homeostasis thanks to these pro-inflammatory pathways which are triggered by senescent cells. Because in principle, if you think for a second, cells have all the means to keep themselves in pristine conditions. Everything that breaks down in a cell can be replaced. You can make new RNA, you can make new proteins, you can even make new organelles. There is one single component in the cell that if damaged cannot be replaced. And obviously, this is our gene. So do we age because we accumulate damage in the only irreplaceable component in the cell, our DNA? Well, the answer would be yes, because indeed there is pretty good evidence that DNA damage accumulates during the process of aging, and it accumulates even more so in pathological conditions associated with aging. So we may have found an answer to this question. However, there is a slight complication, and the fact is that DNA damage is actually repairable. And so how can transient lesion, how can they lead to a persistent condition like senescence? Senescence, it's really a dead end. There is no way back. It's not a transient condition. It's a chronic dead end. Cells will forever be unable to proliferate. If lesions are transient because repairable, the two things really cannot, one transient lesion cannot lead to a persistent, protracted, perennial cell cycle arrest like the senescence condition. So we have to come up probably with a slight modification of the model, and we have contributed to some to generate some evidence in support to an improved version of this model, because we observed that if you take some normal cells and you expose them to ionizing radiations, this generates DNA damage, which is random. And I hope you see my arrow here. You see DNA lesions, which will be random, so they will be scattered throughout their genome, and this will trigger what we call a DNA damage response, so a cellular response to this DNA damage generation. This DNA damage response will, of course, favor the repair of these lesions. However, and that is the discovery, we discovered that those lesions that happen to occur within the tips of the chromosomes, the telomeres, well, those lesions will not be effectively repaired for a number of evolutionary reasons, and therefore those lesions will not be repaired, they will persist, and this will trigger a persistent DNA damage response activation, which is what triggers senescence. So the message here is a simple one. So telomeres with time will become sinks, hubs, black holes of irreparable DNA damage. And DNA damage is constantly being generated in the cell. It's physiologically normal that here and there DNA damage occur. But if this DNA damage is repairable, this again will be unconsequential. But for those lesions that are irreparable, that will lead again to this protracted DNA damage response, this persistent alarm in the cell, which is ringing, and it is telling the cell there is something very important, very serious going on. Again, its genome integrity is affected, is threatened. Probably the involvement of telomere biology in the context of cell senescence and cell aging is not unexpected completely, because we and then others have reported that telomeres, again, the end of the linear chromosomes, will become shorter and shorter and shorter when cells proliferate, and when eventually cells have telomeres which have become too short, this again triggers a DNA damage response, triggers an alarm that leads to the arrest of the proliferation of these cells. And therefore, both in proliferating cells, which is probably the condition here on the right, or in non-proliferating cells, which is an equally relevant condition. I remind you that most of the cells in our body are non-proliferating. Neurons, cardiomyocytes are terminal differential cells. They will not proliferate. Nevertheless, they do age. And we and others have reported that they do accumulate DNA damage during time, during the process of aging, and some of this DNA damage is actually telomeres. So we propose to answer that initial question, what is the trigger of that loss of homeostasis, where we propose that the loss of telomere integrity is that irreversible event that causes cells to age. However, let me take the discussion to the next level up, because it would not probably be completely correct to say that DNA damage causes cellular senescence, because it's the activation of the DNA damage response pathways that mediate the consequences of DNA damage generation, including the establishment of cells. So very briefly, what is this DNA damage response? It's essentially most of the proteins that you see here described in this cartoon that accumulate at sites of DNA damage, and there they fuel the signaling, the presence of DNA damage, and of course, the repair of these DNA lesions. Now, this, and maybe in case you were not familiar here, these are two examples. So these are two cells. Here in blue, you see the nuclei, and you see these dots. We call them foci, and these are individual DNA lesions in which all these proteins have gathered and run to the sites of DNA damage in order again to signal the presence of DNA damage and favor the repair of these DNA lesions. This is the canonical view. We contributed to slightly change this view of the DNA damage response by demonstrating that in fact pretty much everything that I describing in this slide. So the foci formation, the consequences of this foci formation, meaning the transient checkpoint reinforcement and certain cell senescence, would not take place if you do not allow the synthesis and the processing of RNA molecules at the sites of DNA damage. This was unexpected, so we had to become almost an RNA lab, and so we had to, so we suffered through it, and so now I think we have a slightly better understanding than in the beginning of the events, which anyway they are complicated, and probably most of you are not interested in the molecular details, so I've just summarized them in a animated cartoon so I can show you the rest of the data, which are mostly unpublished. So what we discovered that when DNA is broken, this is initially recognized by protein DNA damage sensor complex is called MRN. It's just a protein that detects and accumulates at sites of DNA damage, and that was already known. What we discovered is that this complex loads at sites of DNA damage RNA polymerase II. And it is an active RNA polymerase II that will transcribe bidirectionally what we call damage-induced long-encoding RNA. These RNA are further processed by Roche and Dicer into smaller RNA that then bind back to the unprocessed complementary RNA. And so in short, when you break DNA, you have the synthesis of RNA. These RNA are necessary to allow the formation of this foci by retaining these proteins around sites of DNA damage. Among the many proteins that accumulate at sites of DNA damage, 53BP1 is one of those that we have characterized in more details. Now, how does RNA work? Well, we demonstrated that this RNA allow the liquid-liquid phase separation of these DNA damage response factors. You know how familiar some of you are with this notion. Basically, it's a way to increase the local concentration of some proteins within a defined space, and people also call it condensation. And so, in fact, we demonstrated this DNA damage response foci. They are actually not solid, static, stiff structures, but in fact they are very wobbly, liquid, and viscous, in fact, liquid structure. I'm going to show you some results in some evidence in support of this. So, for instance, if you just look at cells that have been damaged, you see that initially you get this phase in which these foci grow in numbers. So each one of these lesions is triggering the recruitment of these proteins, of this DNA damage response foci. And so you see this white signals here that then they undergo coarsening and fusion, and therefore they reduce in number, so they become larger because they undergo merging between the structures. And this is already some evidence of a liquid structure. They really look like, droplets of oil on the surface of a water solution. But probably even more convincing is to show the behavior of one of these DNA damage response forces. So we took a high-speed movie of 40 seconds in the lifetime of a focus of this DNA damage response properties, and let's see how they behave. As you can see, they are extremely mobile and wobbly, and certainly not static. And this is, again, an additional evidence of their liquid behavior. And probably the strongest evidence in favor of the liquid nature is a FRAP experiment in which you essentially burn the GFP of one of these DNA damage response factors, which has formed this focus, and you see how long it takes to recover the signal within that hole that you have pierced within that focus. And as you can see here, in 10%, you recover already 50% of the signal. So it's an incredibly fast recovery, which is, again, consistent with this liquid nature. Now, this is we have already published a little bit about this liquid nature of these structures, but now we have taken a step ahead in characterizing better these events. And so what we did was to take this protein, 50qubit1, which is one of these proteins that assembles at sites of DNA damage, and we chopped it into smaller bits. We expressed different regions of this protein, and we make them recombinant, we purify them, and then we mix them with increasing amounts of RNA. And as you can see here, there is an individual region of 50qubit1 that is sufficient to form RNA-dependent blobs, these structures, you may call them 50qubit1 foci, in the absence, in this case, of DNA damage, but still in the presence of RNA. So they are RNA-dependent structures. So now we are characterizing many of these events. We are characterizing their properties. We are characterizing which RNA are necessary for their formation. So we think this is quite an informative experimental setup. But let me maybe move to probably what is, I think, conceptually the most important slide here, because what I told you so far is that if you look at this slide here, at this image here, you see all these DNA damage response foci, which are all made by the same protein, like 50qubit1, will go wherever DNA breaks. However, each one of these structures, each one of these dots, each one of these foci is held together by different RNA, because it's an RNA which is generated at each individual genomic site, damage site, because it's upon DNA damage that this RNA is getting synthesized. So the idea is that if you want to inhibit DNA damage response for events here at this site only, you cannot inhibit the proteins, because the proteins will go wherever DNA breaks. So you would not be able to achieve selective specificity. Differently, the only way, if you want to control events at the individual DNA damage response foci, you have to target the RNA, because the RNA is the only specific component in these structures. So the idea is to control the DNA damage response at individual damage sites, not by targeting the proteins, but by targeting the RNA. So the idea is to use an antisense oligonucleotide, an ASO, able to target this RNA, and by binding to them, inhibiting their function, and essentially disassembling, dissolving this DNA damage response foci, hence causing the inactivation of the DNA damage signal. This is the most of the slide, this is a cartoon, but this is actually works, and it works really selectively. So we perform the following experiment. This is a nucleus of a cell in which we can introduce 3 double-strand breaks. They are all embedded within an artificial set of sequences. We call them TET DNA sequences. And in the same cell, we can introduce 2 more double-strand breaks. They are embedded within lac repeat sequences. We can cut off 5 of them, and then we get this chromatin modification event, which is called gamma H2X, which is totally RNA independent. So whatever we will do, gamma H2X stays, and we know, therefore, that now in this cell we have at least 1, 2, 3, 4, 5 sites of DNA damage, which are in this moment presently signaled. Now in this cell we can treat these cells with an antisense oligonucleotides that can target and inhibit selectively the RNA generated at this TET damaged genomic sites. And what we observed that this TET antisense oligonucleotide impedes the accumulation of 50-to-1 at these 3 sites, while in these 2 sites is normally recruited like in any other cell. So this really says that we can control the DNA damage response in a selective manner by using this antisense oligonucleotides. So to recap what I told you before I told you that telomere biology, telomeric DNA damage is the most consequential, the most impactful, because it's the one that is less repairable, so the one which is more persisting. So telomeric DNA damage is extremely dangerous. And then I'm saying that DNA damage sites synthesize RNA that are necessary to activate the DNA damage response, and I can control these events by an antisense oligonucleotide targeting this RNA. So we can now bring the 2 stories together, and we can say that whenever tumors are damaged because too short or somehow damaged, as it happens, they will be triggering a DNA damage response. So you have this DNA damage factors being recruited to these damaged tumors, and their recruitment and their retention at dysfunctional tumor, damaged tumors, is really dependent on the RNA which are generated at the sites of DNA damage, in this case, RNA generated at the tumors. And of course, they have the telomeric sequence, which is a known sequence. We know that tumors have the TTA-GGG sequence. So now we can design an antisense oligonucleotide targeting this telomeric RNA, and this telomeric antisense oligonucleotide will bind to this RNA and therefore disassemble, disengage, dissolve this DNA damage response foci, effectively working as an inhibitor of the DNA damage response, but selectively at the tumors. And therefore, we used this approach in many different settings. Before I show you some evidence in cells with the LAC and TET systems, this approach works also in tumor cells. But I'm going to straight into the evidence of its efficacy in vivo, which I think is the most relevant one. So we used mice in which we can induce telomeric dysfunction by removing a protein called the TRF2. TRF2 is a protein that sits on the tumors and protects them. As soon as there is no TRF2, now these tumors are exposed DNA ends, and they trigger the DNA damage response. So as soon as we remove TRF2 genetically, we also treat these mice with an antisense oligonucleotide. This is a systemic delivery. It's a needle in the belly of these mice, and this is unassisted. And what we observed that now these mice that in their tissues, so these are not cells, these are tissues from these mice. These tissues that mount a DNA damage response, with, as you can see in these 2 independent markers, where this DNA damage response is strongly reduced in mice lacking treated with this antisense oligonucleotide. So this proves that this antisense oligonucleotide approach works quite effectively in controlling the consequences of telomeric DNA. So for the first time, in fact, we have a way to uncouple DNA damage from its consequences, the DNA damage response activation. And therefore, we have a tool to answer the question, the phenotype that I'm studying, the pathology that I'm modeling in this animal model or in this cell system, is the disease contributed in any meaningful way by tumor dysfunction? Well, now we have an inhibitor, a selective inhibitor, that if added to the system can tell you whether that condition, that phenotype, that situation was dependent on dysfunctional tumors and the activation of the DNA damage. So now we are using this tooling in many different settings. And just to give you an example, so what we published was it's using a very complex condition. It's an accelerated aging syndrome known as Hutchinson-Gilford progeria syndrome, in which many things go wrong. You have a nuclear shape which is dysfunctional, you have a loss of heterochromatin, and you have also tumors which are which are shortened and damaged. And so, is this telomere phenotype making any significant contribution to this disease, for instance? And we treated mice that carry this mutation selectively in the skin for a number of technical reasons. And what we observed that now these mice can live nearly 50% longer when treated with this antisense oligonucleotides. So this answer the question, is tumor biology contributing to this disease? Yes, because if you inhibit the consequences of tumor dysfunction, you extend the lifespan. Of course, the disease is not just all about that, because these mice still live shorter than normal one-time mice. Luckily, HGPS is a rare disease. It's a rare disease, but there are in fact many diseases in which tumor dysfunction has been characterized. And here we have summarized in this review that we published nearly 1 year ago about the accumulation of dysfunctional tumors and the consequence of tumor dysfunction, meaning DNA damage response activation of the tumors and the accumulation of senescent cells in different very relevant human conditions. And maybe I'll, on this occasion, since I understand that most of you are interested in liver conditions, I have added one slide which summarizes some of the evidence in regard the contribution of tumor dysfunction to liver diseases. And again, in liver cirrhosis, that probably I don't need to describe to most of you. Well, in patients, in cirrhotic patients, a number of them have been shown to have a telomerase mutations in a percentage, with an incidence which is higher than control patients, and this leads to short tumors. We also know that NAFLD as in the liver, these patients show what we call TAF, for tumor-associated DNA damage response forces, or DNA damage response foci that are localized in the tumors. That means they have damaged tumors, and other markers like p21 that really correlate with the senescence conditions triggered by telomeric dysfunction, and their accumulation correlate with the severity of the disease. And there are also other conditions like BBC, which is also associated with shortened tumors and markers of DNA damage response activation and senescence. So again, there is certainly a good evidence linking tumor dysfunction to liver diseases. However, probably among the human, many human conditions that we have characterized, probably the one that showed the most convincing link, and actually a causative link between tumor dysfunction and this human condition, well, that was in the context of idiopathic pulmonary fibrosis, lung fibrosis. This is because IPF patients display invariably markers of DNA damage response activation in the tumors, they accumulate senescent cells, and they have this suspect phenotype, this pro-inflammatory cytokine phenotype. Invariably, patients have short telomeres, and you can see this both in their PBMCs, but also in their, of course, in their lungs. And if you look, if you stratify individuals according to their telomere length, well, 1/3 of the familiar cases or 1/4 of the sporadic cases have very, very short telomeres, meaning below the 10th percentile. Meaning, again, they are extremely, they are patients with extremely short telomeres. And consistently in the independent cohorts, and the results keep coming up robust and robust, short telomere length is really a bad idea in terms of response to therapy, progression to the disease, and even upon lung transplantation, patients with short telomeres are those that respond more poorly. And probably the most convincing evidence of a causative role of telomere dysfunction in this disease is the evidence that, again, many familiar but also sporadic cases of IPF patients carry mutations in telomere biology genes, including telomeres. And therefore, when we observed that, we thought maybe genetic model, animal model of IPF could be useful to adopt in our lab. So we adopted it. So we use a telomerase knockout mouse. So this is a mouse strain in which the gene that maintains telomeres long, the telomerase gene, has been inactivated. So we adopted that. And then we treated these mice with our antisense oligonucleotides. And then we monitored the impact that this had at different time points up to the very last one, which was at nearly 1 year of age. And what we observed is that first, we observed that these mice, which have short telomeres, they also activated DNA damage response, which is to a good extent reduced very significantly, actually, significantly reduced in mice treated with the 2 independent antisense oligonucleotide. This has consequences in terms of the histology and the structure of the lungs. You see these holes, these areas in the lungs which are pathological, which are reduced in number and size in mice treated with these antisense oligonucleotides, and you can see here. And when you perform an RNA-seq experiment, so you look for pathways which are dysregulated in these mice, well, you observed that these mice have a high induction of pro-inflammatory pathways, which are reduced significantly when these mice have been treated with this antisense. So this is particularly relevant because pro-inflammatory pathways and inflammation, chronic inflammation, is actually what causes fibrosis, which of course gives really the name to this disease. So we tested fibrosis in these mice, and what we observed that these mice, that when we start treating them, these are the knockout mice, and these are the wild-type mice, again, staying with this technique that allows you to detect fibrosis in the lungs. Well, these mice show this level of fibrosis, which is already higher than wild-type mice, and this disease is a progressive one in humans and also in mice. And so with time, as you can see, these mice accumulate more and more fibrosis, both when they are untreated or treated with an antisense oligonucleotide, which is not effective. However, when mice have been treated with these 2 independent antisense oligonucleotides, we strongly reduce the fibrosis in these mice, and so we can probably safely say that we can stop the progression of the disease. So this disease that was progressing in this direction toward higher fibrosis is actually stopping this increase. And actually, not only stopping the increase, it is probably also reverting to an extent, at least it seems, the fibrotic process, because here we are reaching a lower level, for sure also here, a lower level in this treated mice. So I think there is a potential for this approach, not only in mice, but hopefully also in humans. Let me now very quickly go through some additional evidence suggesting that we see an impact not only in the lungs, but also in the immune system, in the hematological compartments, so both in the bone marrow and in the spleen. You see, in induction of pathologies which are reduced in mice treated with these antisense oligonucleotides, and this applies not only to different kinds of cell types that I will definitely not go through all of them because I don't want to bore you, but what probably I think is particularly exciting to me is the observation that the stem cell biology, which is affected by the lack of telomerase, is also rescued instead by the treatment with these antisense oligonucleotides, as we are observing here. And I will conclude this part by saying that yes, we see mouse as something that is extremely informative for the human condition, but it's not the only one. There are other animal models that can be exploited. One of them is zebrafish. Zebrafish can have tumors of the same tumors of the same size of humans, and there are zebrafish lines which have mutations in the telomerase gene, and also here what we observe in two independent collaborating lab that the survivance for the longevity, but also the fitness of these species is improved by reducing the DNA damage response activation selectively at the telomeres, again with this antisense oligonucleotide. So this is what really I wanted to tell you a little bit about aging related conditions, but very briefly, I mentioned before that aging is one of the highest risk factors for cancer, and therefore we thought whether there was also an opportunity to impact on cancer with our approach. Now, again, always targeting the tumors. Why tumors? Because there is a probably not very popular, not very famous type of tumors in which telomere maintenance is not maintained by telomerase re-expression, which is the common way tumors acquire immortality. They want to avoid this telomeric shortening problem by re-expressing telomerase. And this is again what most tumors do. But some others, however, instead what they prefer to do is to induce a mechanism known as ALT, for alternative lengthening of the telomeres, in which basically telomeres are constantly damaged, constantly in need of repair, and this repair is a sort of homology recombination-based repair. And again, in this case, the DDR, the DNA damage response, is something that is vital for these tumors, because it is necessary to maintain the telomeres and therefore necessary to maintain the viability of these tumor cells. So then the idea that immediately ahead, of course, is to target these tumors with our approach. But of course, first thing, do these tumors tend to have higher levels of this telomeric non-coding RNA? And the answer is yes, if you measure them by RT-PCR in this case, both on one strand, the G-rich strand, so telomeres are G-rich, they have a G-rich strand and a C-rich strand, so we tend to call G-rich and C-rich RNA. And as you can see in red, you see the ALT tumors, in blue the non-ALT tumor, you see the red ones typically have a higher levels, and if you have also matched or semi-matched cell lines, you see that consistently ALT tumors are stronger in the expression of this RNA marker. And of course, then the obvious experiment is the one that I mentioned before. So you use to treat these cells with antisense oligonucleotides targeting this RNA and reducing the viability of these cells. And what we observe that now these AIRC cancer cells stop proliferating, while other normal telomerase positive cells or normal cells are absolutely unaffected. And this is really a cytotoxic event, is not cytostatic, because you can see that we are inducing here a DNA damage response. I will certainly not go to the details of the intricate molecular biology pathways necessary to maintain tumors in alt cancer cells, because it's extremely complicated. But I'll just summarize our conclusion here. We observed that with this antisense oligonucleotide approach, we observed the reduction of the accumulation of DNA damage response factors at the tumors. Cells, alt cells try to survive by triggering an alternative response to that, and this is, however, not effective, and therefore the cells keep accumulating DNA damage as identified by this approach, and this triggers, in fact, the death of this cell. So we are proposing this as a way to control the survival of cancer cells, in this case, induce their death by targeting telomere maintenance mechanisms, again, by targeting the RNA being generated at these dysfunctional teams. So to summarize things, we think that we identified teams as the ultimate vulnerability in the cells, because they are the irreparable portion of the irreplaceable component, which is really our genome. And we have identified a way to inhibit the consequences of this DNA damage response activation, also selectively at the tumors. So this is where I usually end my seminars. I just want to give you really a very super short summary of something that we have reported recently, and I will probably let me, because I don't want to spend too much time here. I just want to show you this evidence that appeared in the literature, and was that you may have heard that SARS-CoV-2 infection, COVID-19 seem to accelerate aging in some ways, and this was also associated with the induction of cellular senescence. And this was reported by many reports. You can see here the evidence of some of those publications, but the mechanisms that were triggering this DNA damage response, so this cellular senescence process were not clear. And since we are a DNA damage lab, we really thought that maybe DNA damage was involved. So what we did was to start a collaboration with Alessandro Barcello in ICGB in Trieste to test the impact of the infection on normal human cells with of viral infection. And what we observe, and I appreciate here, there are very many results and very many Western blots and immunofluorescence results and their quantification. I make it simple. We observe that, in fact, this virus, when it infects human cells, breaks their genome, so causes DNA damage. And we also identified the pathways, but again, this is not really the topic of my presentation today, so I'll go straight to the conclusions out of the story that, again, came out a few months ago. So what we discovered, that when SARS-CoV-2 infects cells, this leads to the degradation, to the loss of one important factor in the cell, which is called the CHEK1, and this leads to a loss of the NTPs, and therefore without the NTPs, cells cannot proliferate, and this triggers a DNA replication problem. So on one side, the virus breaks DNA, but as I mentioned before, DNA is repairable. So if it is repairable, how can this lead to cell aging, which is irreversible? Well, what we discovered that, in fact, not only the virus breaks DNA, but also impedes the recruitment of 53BP1, which is the protein that I was discussing before, which helps DNA repair. And so we observe that different mechanism of 53BP1 is inactivated, and therefore there is a DNA repair defect. And so combining a DNA repair defect and a DNA replication induction situation, you can only come down with the accumulation of DNA damage, and of course the establishment of cell senescence and the triggering of the pro-inflammatory pathways. And many of you will remember the cytokine storm that many people have described upon a SARS-CoV-2 infection. We believe that certainly there is a component, an immune-mediated component to that cytokine storm, but we think also that the cytokines secreted by senescent cells, senescent because infected by the virus, we think that that is also an additional mechanism that leads to this strong inflammatory response. So I stop here. This is us, and that was, when we celebrated 20 years of my lab here in Milan. And of course, we are constantly hiring a talented PhD student and postdocs in case any one of you is interested. So I'll stop here, and I'll be very happy to take your questions.
Well, thank you very much for this exciting presentation. Really nice way to finish our series.